Assalamualaikum and a good day to everyone. Today we are from Group 6 would like to present about Practical 1, which focus on the effect of environmental factors on the growth of microorganisms. So, microorganisms like bacteria and fungi do not grow randomly. They are highly influenced by their surrounding environment. Each microorganism has specific conditions in which it grows best. There are several key environmental factors that affect the growth. Firstly, temperature. Temperature will affect how fast the microorganism grows because it influences the enzyme activity. Each microorganism has its minimum, optimum, and maximum temperature for growth. Secondly, osmotic pressure that affect the movement of the water in and out of the cell. When the surrounding environment has too much solute, water leaves the cell, which can slow down the growth. On the other hand, if there are too little solutes, too much water can enter the cell and can cause damage. Next, pH also plays an important role. Most microorganisms grow best in neutral condition, but some of it can survive in acidic or alkaline condition. Lastly, oxygen availability. Oxygen availability determines the type of microorganism that can grow. Some require oxygen, which is called aerobic, while the others grow in the absence of oxygen, which is called anaerobic. Understanding these factors is important in areas such as food preservation, safety, and also microbiology. So, the objective of these experiments are to study the effect of temperature on the growth of microorganisms, to examine how osmotic pressure influences the microbial growth, to investigate the effect of pH in microorganisms, and also to observe the role of oxygen in the growth of different microorganisms.
For the method of salt and sugar, add a 0.1 ml of the same culture into tubes containing different concentration of salt and sugar to test the effect of osmotic pressure. For the effect of pH, add 0.1 ml of the same culture into 4 TSB tubes adjusted to pH 3, 5, 7, and 10. To test the effect of oxygen, add 0.1 ml of the same culture into 2 TSB tubes. In one of the tubes, add a thin layer of paraffin oil on the surface to block oxygen. All of these are then incubated in 37 degrees Celsius for 24 hours. And finally, for the temperature, add 0.1 ml of the same culture into 4 tubes of TSB and incubate them at different temperatures: 5 degrees Celsius, 25 degrees Celsius, 37 degrees Celsius, and 50 degrees Celsius.
The next growth factor of E. coli are E. coli in salt solution. There are 4 different concentrations that has been checked, which is 0%, 1%, 5%, and 10%. In 0% salt solution, there is a good growth. This is because there is no glycolysis happen and there is no osmotic stress. Thus, the E. coli can grow more steadily as it is in the range of its optimal temperature, which is 37 degrees Celsius. In 1% solution, E. coli growth is moderate. This is because its growth decreased from 0% because there is an increasing concentration, which causes its plasmolysis start to happen and the osmotic stress. There is also osmotic stress. E. coli. This causes E. coli growth inhibition to increase. That is why its amount is also decreased. In 5% salt concentration, E. coli has poor growth. This is because the salt concentration is decreasing significantly, which causes its growth inhibition to increase significantly. In 10% salt concentration, there is no E. coli growth because it has high salt concentration, which causes it to inhibit the E. coli growth overall. In conclusion, E. coli can only grow in environment that salt concentration less than 1%. But even though E. coli cannot grow in high salt concentration, there are other bacteria that is halotolerant, like Bacillus species and Staphylococcus aureus. This is E. coli in sugar solution. We check with 4 different concentration, which is 0%, 1%, 10%, and 20%. In 0% sugar concentration, there is moderate growth. This is because there is limited nutrient, which causes the E. coli need to use the nutrients efficiently. In 10% salt concentration, there is moderate growth. This is because there is limited nutrient, which causes the E. coli need to use the nutrients efficiently. In 20% sugar concentration, there is moderate growth. This is because there is limited nutrient, which causes the E. coli need to use the nutrients efficiently. there is no osmotic stress happening. In 1% sugar solution, there is good growth. This is because it has moderate glucose concentration, which can enhance the E. coli growth significantly. It also has the amount of nutrient that can support the growth of E. coli. That's why the E. coli growth increase. In 10% sugar concentration, there is no growth of E. coli. This is because it is high in sugar concentration. Thus, it may cause the E. coli to dehydrate and cause significant osmotic stress. It also causes the E. coli metabolism to be disrupted, cause it to be dead. 20% sugar concentration, it also has no growth. This is because it is high in sugar concentration. And second, its metabolism was also disrupted, thus inhibit the growth of E. coli. E. coli cannot grow in a really high sugar concentration in environment with really high sugar concentration. But there are also other bacteria that can grow, like bacillus and lactic acid bacteria, which commonly found in fermented foods. Lastly, we will discuss about the E. coli growth in the presence of oxygen or when there is no oxygen available. When there is oxygen available, E. coli has good growth because E. coli primarily use aerobic respiration. Thus, it has higher activity and efficient energy production. When there is no oxygen available, E. coli has poor growth because it needs to switch to mixed acid fermentation. This will cause the energy production to be less efficient because it uses substrate level phosphorylation.
Now I will explain about how pH and temperature factors can affect the growth of E. coli. E. coli is a mesophilic bacterium, which means it grows best at optimum temperature. First, let's look at the temperature factors. At 5 degrees Celsius, there is no growth. This is because cold temperature will cause the cell membrane to become rigid. Next, at 25 degrees Celsius, we observe there is poor growth. This is temperature. This is because the temperature is below the optimal temperature, so metabolic activities occur more slowly. At 37 degrees Celsius, E. coli shows good growth. This is because 37 degrees Celsius is their optimum temperature, so the enzyme function is at their highest efficiency. Lastly, at 50 degrees Celsius, there is no growth. This is because high temperature can cause protein and enzyme to denature. Next, let's move on to pH factors. At the first pH, which is the pH 3, there is no growth. This is because the highly acidic condition will cause the protein denature and the ATP synthesis to stop. At pH 5, there is poor growth. The acidic environment will create stressed environment for the cell. At pH 7, we see good growth. This is because pH 7 is the optimal pH for the E. coli bacteria. As a result, nutrient uptake and metabolic processes occur more rapidly. Lastly, at pH 10, there is no growth. The highly alkaline condition causes protein to denature and interfere with the cell ability to maintain its shape. As a conclusion, E. coli grows best at 37 degrees Celsius and pH 7, while extreme temperatures and extreme pH will inhibit and completely stop their growth.